Hello everyone, welcome to the next lecture in our physiology video lecture series. This lecture is titled Blood Cell and Immunity. In this lecture, we will go through the structure and function of red blood cells and white blood cells as well as platelets, and also learn how they function. We will also go through the concept of immunity, looking both into innate immunity and acquired immunity from the perspective of blood cells. So let us begin. To start off, we will start off with red blood cells. Red blood cells are also known as erythrocytes. As we know, they are the major carrier of oxygen from the lungs to different tissues in our body, and they use the protein hemoglobin to do it. This is the major function of RBCs that we know. There is another major function, which is to act as an acid-base buffer. So, in addition to hemoglobin, RBCs also contain large quantities of carbonic anhydrase, an enzyme that catalyzes the reversible reaction between carbon dioxide and water to form carbonic acid. Thus, this carbonic anhydrase makes it possible for the water of the blood to transport enormous quantities of carbon dioxide in the form of bicarbonate ion from tissues to lungs. So, in one direction, RBCs take oxygen from lungs to tissues in the form of hemoglobin and also help in carrying the excreted carbon dioxide from the tissues to the back to the lungs in the form of carbonic acid. The use of carbonic acid also plays a role in acid-base balance of the blood. So, as we know, the pH balance of the blood is very finely maintained around pH 7.5 to 7.8. Once this pH is shifted to either side, excess amount of carbonic acid can be made can be delivered to the lungs to be exported out of the lungs in order to increase the pH, or the opposite can also be done in order to maintain the pH of the blood at the optimum level. So these are the functions that the RBCs do. What are the various characteristics that RBCs have? Red blood cells are enucleated, that is, they do not possess a nucleus and cannot replicate. So RBC is a terminally differentiated cell that cannot replicate further. The RBCs have a biconcave structure, that is, they are concave from both ends, as we can see in this picture, and have a disc-like appearance with a diameter of 7.8 micrometer and a thickness of 1 to 2.5 micrometer. Further, due to the absence of nucleus or any big organelles present inside the cell, the RBCs can squeeze through small capillaries as well, where no other cells can cross through and deliver oxygen to those tissues present beside the capillaries. The RBCs have a volume of around 90 to 95 cubic micrometers, but there are also a huge number of RBCs present in a cubic millimeter of blood, range which is around 5 million RBCs. So there are around 5 million RBCs in 1 cubic millimeter of our blood. This is the standard, and ordinarily in males the amount of RBCs are a bit higher, and females it is a bit lower than this mark. Further, the RBCs are also characterized by the hemoglobin content that they carry, and it is around 14 to 15 grams per 100 ml of blood. So that was about the characteristics and function of RBCs. Let us look into what is the origin of RBCs. In the early yolk sac, nucleated red blood cells are produced by the yolk sac. In the early embryonic life, the nucleated red blood cells are produced by yolk sac. So these are not mature RBCs, but they have the nucleus. During the middle trimester of gestation, liver is the main organ which produces RBCs, but reasonable numbers are also produced by spleen and lymph nodes. Spleen and lymph nodes are lymphatic organs that are present in our body. During the last month or so of gestation, the RBCs are produced exclusively by bone marrow, and this bone marrow is the site of production of RBCs for the rest of our lives. The blood cells begin their lives in bone marrow from a single type of cell called a pluripotential hematopoietic stem cell with self-renewal. So here we have to understand a lot of things. First, the first thing I want to tell you is that what are stem cells? Stem cells are early progenitor cells that have the capability of self-renewing and producing different progenitor cells which then produce different cell types. The hematopoietic stem cell is one of the pluripotent stem cells that is present in the embryo as well as the adult body, which can generate all the blood types or blood cell types in our body. So one cell can give rise to all the blood cells, which include the RBCs, the lymphocytes, the and the white blood cells and platelets as well. Further, since it is a stem cell, the hematopoietic stem cell can self-renew. That is, when it makes two copies by binary fission, it can make another stem cell which is exactly identical to the parent and another daughter cell, which then can further differentiate to form other blood cell types. So the pluripotent hematopoietic stem cell is the one that gives rise to the whole blood cell types. Further, a committed stem cell that produces erythrocytes is called a colony forming unit erythrocyte. So here is another concept. So stem cells can be divided depending on their potency into different parts. So what is potency? Potency is the ability of a stem cell to form a particular lineage or a particular type of cells. So mainly they are divided in, we can divide them into totipotent cells, which is the zygote itself, which can produce every cell in the body, including the placenta. Pluripotent cells, which can form all the cells of the inner cell mass and the embryo, but not the placenta. Multiprotein cells, which can produce many cell types, but not all. So it is restricted in the ability of the cells that it can produce. And after multipotent, we have unipotent cells, which can only produce one type of cell. So as here we have a pluripotent cells, hematopoietic stem cells are pluripotent cell. But after differentiating a number of times, the daughters that are formed can form restricted stem cells, which can renew themselves, but have a limited amount of time till they differentiate to form terminally differentiated cells. What this means is that hematopoietic stem cells give rise to particular cells such as the colony forming unit cell, which then forms various blood cell types. So the pluripotent hematopoietic stem cell gives rise to the colony forming unit spleen. This is a cell which again can differentiate to form the colony forming unit blast, followed by the colony forming unit erythrocyte, which finally gives rise to the mature erythrocytes. Further, the colony forming unit spleen can also form the white blood cell lineage via colony forming unit granulocyte and monocyte, or platelets via the colony forming unit megakaryocytes. Further, as we know, as I just told you, that the hematopoietic stem cells can also give rise to lymphocytes, which are mainly associated with the immune system via the lymphoid stem cell. The growth and reproduction of different stem cells are controlled by multiple proteins called growth inducers such as interleukins. So the presence of interleukins is what determines if a stem cell will differentiate into a particular lineage. There is another set of proteins called differentiation inducers, which also does the same. So just another piece of information. A stem cell can be of two types. Quiescent stem cell, which does not divide much and stays as a stem cell for most of its life, and activated stem cells, which divide and differentiate into different types of stems, different type of progenies. So depending on the inducers that are given to a particular stem cell, it will want to proliferate to produce more stem cells, or it can be given the signal to differentiate in order to form new cell types. And all of this depends on the signals that are given to the stem cells. And such signals include growth inducers and differentiation inducers. So let us get back to RBCs. And here we have a lineage of how our mature erythrocytes are generated from the proerythroblast. The first cell after the colony forming unit erythrocyte that can be identified as a red blood cell is the proerythroblast. Once the proerythroblast has been formed, it eventually generates the basophil erythroblast, which is known as basophilic because it can be stained with basophilic dyes or basic dyes, but they do not contain any hemoglobin. Followed by that, after a lot of divisions, we get the reticulocyte, which contains still some amount of basic material, and hence can be stained by the basophilic dyes, and still contains remnants of Golgi apparatus, mitochondria, and a few cytoplasmic organelles. Finally, this reticulocyte, after 1 or 2 days, finally removes all its basic material present in it and forms the mature erythrocyte. This is the differentiation of all the RBCs from the proerythroblast to finally the erythrocytes. On the right hand side of the figure, we have examples of red blood cells that have undergone differentiation but with some defect, which has caused them to inherit a disease. So these include microcytic hypochromic anemia. So anemia is a disease of the red blood cells. Microcytic hypochromic anemia means the RBCs are too small to carry anything. Sickle cell anemia is very famous, as we know. And this particular mutation in the hemoglobin protein, hemoglobin gene, causes the red blood cells to take a sickle like appearance. So they get flattened in a crescent shape and cannot carry oxygen well. Similarly, we have megaloblastic anemia and erythroblastis fetalis, which are also two types of defects that are seen in red blood cells. Next, we move on to the regulation of production of RBCs. So the total mass of RBCs is regulated by the fact that there are an adequate number of RBCs that are always available to transport oxygen from the lungs to the tissues. This is a major factor that controls the amount of RBCs present in the blood. Secondly, the cells also have to be at a certain amount so that they do not impede the blood. So as I said, in very small capillaries, the RBCs can squeeze through and pass the capillaries while delivering oxygen to the tissues around it. But if there are a lot of RBCs present, then they can impede or jam these small capillaries, and thus oxygen transfer to that particular tissue may be inhibited. In such a case, the amount of RBCs generated are lower, so that such a blockading or impeding of the blood flow does not occur. Tissue oxygenation is the most essential regulator of RBC production, as is obvious, because that is the major function of the RBCs. Any condition that causes the quantity of oxygen transported to the tissue to decrease ordinarily increases the rate of RBC production. So in case there is less amount of oxygen reaching the tissue, the rate of RBC production increases. The particular protein that controls all of this is known as erythropoietin. The principal stimulus for RBC production in low oxygen state is a circulating hormone, which is known as erythropoietin. Erythropoietin is released by cells in the kidney and is what majorly controls the rate of RBC production. Erythropoietin stimulates red blood cell production and its formation by the kidneys in the case in response to hypoxia. Hypoxia is the condition where low oxygen levels are encountered by the body. So here we have the flow chart of how exactly this happens. In case there are some factors that decrease oxygenation, such as low blood volume, anemia, low hemoglobin, poor blood flow, or pulmonary disease, all of which decrease the oxygen transported to the tissues. There is a decrease in tissue oxygenation. This decrease in tissue oxygenation signals to to the kidney, which produces excess amount of erythropoietin. This excess amount of erythropoietin reaches the hematopoietic stem cells and allow them to differentiate faster to form the proerythroblasts, which finally form the red blood cells. When enough red blood cells have been formed, thus the tissue oxygenation levels have been increased to normal amounts. This in turn again decreases or down regulates or produces a negative feedback on erythropoietin and lowers the amount of erythropoietin, so that the amount of RBCs can be maintained and there are not a lot of RBCs in the bloodstream. This is how the regulation of RBCs are done, or the control of production of RBCs are done. Next, we move on to formation of hemoglobin. We know hemoglobin is the chief chemical the chief protein complex that acts in order to carry oxygen from the lungs to the tissues and is a chief constituent of the RBCs. But what exactly is hemoglobin? Hemoglobin is a polypeptide which has iron bound to it. There are two parts to the hemoglobin. There are two parts to the hemoglobin molecule. One is the polypeptide part of it, and one is the heme part of it. So the heme part of it is first made of two succinyl CoA, which bind to two glycine molecules to form a pyrrole moiety. Four pyrrole moieties come together to form the protoporphyrin 9 complex. Protoporphyrin 9 then binds to iron, but iron in the plus 2 ionic state to form heme. Heme binds to the polypeptide part of hemoglobin to form a hemoglobin chain, which can be either alpha or beta. Two alpha chains of hemoglobin bind to two beta chains of hemoglobin to form the final hemoglobin protein. So here we have the full structure of hemoglobin, and we can see that the iron is bound to two hemoglobin. Iron is bound for the heme part of in this particular picture. We can see the full diagrammatic representation of hemoglobin, and here we see that the iron is bound in between the protoporphyrin moiety, which is then collected to the polypeptide chain. One thing that we have to keep in mind is that oxygen does not bind to the two positive charges on iron with the help of an ionic bond, as can be intuitive. Instead, it binds loosely with a coordination bond with the iron molecule, iron atom. This is an extremely loose bond and so can be easily reversible. Further, oxygen does not become ionic oxygen, but is always carried as molecular oxygen to the tissue. That is in the form of O2. And as the bond between iron and oxygen is very loose, it is released into the tissue fluid still in the form of molecular oxygen when it reaches the tissues. Next, we move on to destruction and lifespan of RBCs. RBCs that circulate the blood normally circulate for an average of 120 days before being destroyed. The older RBCs have, after circulating a lot in the blood, have become fragile due to age, and the RBC cell can actually rupture during passage through a tight spot in circulation. Such tight spots are present in various organs, especially in the spleen, where red cells self-destruct as they try to squeeze through the red pulp of the spleen. And the spleen is known as a site where older RBCs are destroyed. But when what happens when the RBC is destroyed? The hemoglobin part is phagocytosed immediately, as hemoglobin can be toxic for the brain. And thus it is immediately phagocytosed by macrophages in the liver and spleen and bone marrow, so that it is not released into the blood. Next, the iron portion, only the iron portion of the hemoglobin is released into the blood, and the porphyrin portion of the hemoglobin is converted by macrophages through a series of stages into the bile pigment bilirubin, which is released into the blood, and later removed by the liver via bile. So the bile acids that go into our stomach are actually carrying this bilirubin from destroyed RBCs. So you might have heard of this bilirubin, because in case of jaundice, where we know that the patient that is afflicted with jaundice, his eye color turns yellow and his skin also starts turning yellow. This is a direct result of a release of bilirubin into his bloodstream. The amount of bilirubin that is released into the bloodstream due to destruction of RBCs increases in case of jaundice. And bilirubin, when as it is a pigment, it stains different internal organs with a yellow color, and thus people with jaundice have this yellow coloration on their skin and eyes. So that ends our discussion with RBCs. Next, we move on to blood leukocytes, granulocytes, and monocyte macrophage system. So we have moved on to white blood cells. And as we know, white blood cells are immediately responsible for protecting our body from foreign organisms. Our body has special system for combating different infectious and toxic agents. This is comprised of blood leukocytes, that is the white blood cells, and tissue cells that are derived from these leukocytes. These cells work together in order to prevent disease by actually destroying the invading bacteria or viruses by phagocytosis. So if you look back, we have discussed phagocytosis before. It is the total engulfing of a foreign particle by a leukocyte, or by forming antibodies and sensitized lymphocytes. So antibodies we will also look into, which are proteins that actually bind to foreign antigens, and both of these may work in order to either completely destroy the invading organism or inactivate it. So we have a similar diagrammatic representation of genesis of myelocytes. So to start with, the starting cell this time is known as a myeloblast, which then converts into a promyelocyte, which gives out a lot of progenies. The first is the megakaryocyte, which then further divides in order to form platelets. The myelocyte also divides into neutrophil myelocyte, which after successive divisions gives rise to the neutrophil. The eosinophil myelocyte, which after successive divisions give rise to the eosinophil, and the basophil myelocyte, which after successive divisions give rise to the basophil. And finally, these on the right hand side, this lineage is the monocyte lineage that finally forms the monocyte, which are essentially immature macrophages. We will learn about all of this in detail. This, as we have seen, leukocytes also have another branch, which are the lymphocytes, the T cell and B cell lymphocytes that are essentially associated with immune system of our body. So what is the lifespan of WBCs? The lifespan of granulocytes, that is the All these cells, the neutrophil, the eosinophil, the basophil, and the monocyte, are all known as myelocytes or granulocytes. They are known as granulocytes because they have small granules, as we can see right here in the pictures, present in the cytoplasm. These granules are filled with the digestive and cytotoxic enzymes, as well as the cell membrane damaging enzymes such as perforins, which are released onto the invading organism in order to destroy it. So the granulocyte, after being released from the bone marrow, is the lifespan of the granulocyte after being released from the bone marrow is normally 4 to 8 hours circulating in blood, and another 4 to 5 days in the tissues where they are needed. The monocyte also have a short transit time, 10 to 20 hours in the blood, before wandering into the tissues to become macrophages. Macrophages are essentially mature forms of monocytes. Lymphocytes have lifespans of some weeks to a long time, such as months. And the longest lasting of the lymphocytes are the memory lymphocytes, which keep record of the infection that has been faced by the body. And finally, platelets in the blood are replaced once every 10 days. So platelets just have a high turnover and are replaced very fast. So let us look at neutrophils and macrophages and how they act. It is mainly neutrophils and tissue macrophages that attack and destroy invading bacteria, viruses, and other injurious agents. Neutrophils and monocytes can squeeze through pores of the blood capillary by diapedesis. So diapedesis is the process by which the neutrophils and macrophages present in the blood can circulate directly into the tissue. So this side is the tissue side. So one thing we have to understand that most infections occur in a particular tissue, and our neutrophils and macrophages and every leukocyte that is present are present in the blood. So how do they reach the site of infection? What they do is they, after arriving close to the site of infection, they actually start to squeeze through the basement membrane of the capillary cells. And after squeezing through, they are able to push through into the tissues themselves and start moving towards the source of the infectious material or foreign object. Both neutrophils and macrophages can move through tissues by amoeboid movement. Some cells can move with velocities as great as 40 micrometers per minute, which is equivalent to their whole length per minute. That is a huge speed for such small cells. Finally, many different chemical substances in the tissues, such as bacterial toxins or products of complement complex, cause both neutrophils and macrophages to move towards the source of the chemical. This phenomenon is known as chemotaxis. So, the foreign bacteria or the foreign organism releases some kind of toxins or some kind of chemicals, or the body's own cells in response to these foreign cells can release some kind of chemicals that actually attracts neutrophils and macrophages to the site of infection. Such a substance is known as a chemotactic substance, and the whole process via which the neutrophils and macrophages travel through the blood into the tissues towards the source of the chemotactic agent is known as chemotaxis. So this is how neutrophils and macrophages work. So we'll look into the phagocytosis by neutrophils and macrophages a bit more deeply. And as we know, phagocytosis is the cellular ingestion of offending agent or the foreign object. So essentially, the neutrophils and macrophages essentially engulf the foreign object and digest it inside themselves. But there are some differences between phagocytosis done by neutrophils and phagocytosis done by macrophages. So what are those? On approaching a particle to be phagocytized, the neutrophil first attaches itself to the particle and then projects pseudopodia in all directions around it. The pseudopodia meet one another on the other side and fuse. So essentially, like tentacles, the pseudopodia cover the foreign object and then fuse together to enclose the whole foreign particle inside a chamber inside a chamber which is present in the neutrophil itself. The chamber then invaginates to the inside of the cytoplasmic cavity and breaks away from the outer cell membrane to form a free-floating phagocytic vesicle, which is also known as a phagosome, inside the cytoplasm. So, and the neutrophil can thus phagocytize 3 to 20 bacteria before it becomes inactivated and dies. So, if you remember the first lectures where we talked about phagocytosis, the whole process of ingestion occurs after the phagosome is formed. After a phagosome is formed with the particular foreign objects and bacteria present in it, vesicle that we known as the lysosome, which contains various digestive enzymes as well as other reactive oxygen species which can destroy bacteria and are cytotoxic in nature, fuse with the phagosome. This fusion allows all these cytotoxic enzymes to come in contact with the foreign particles and degrade and destroy them. This is how the process of phagocytosis works. So this is generally how phagocytosis works. So this was phagocytosis by neutrophils. What about phagocytosis that is committed by the macrophages? When activated by the immune system, they are much more powerful phagocytes than neutrophils and are capable of phagocytosing as many as 100 bacteria. So 5 to 10 times more bacteria than neutrophils can phagocytose. They also have the ability to engulf much larger particles, even whole red blood cells or occasionally malarial parasites. Whereas neutrophils are not capable of phagocytosing particles much larger than the bacteria. Also, after digesting particles, macrophages can extrude residual products and often survive and function for many more months. Thus, macrophages are much more efficient phagocytosis cells than neutrophils themselves. Next, we move on to the monocyte macrophage cell system. The total combination of monocytes, mobile macrophages, and fixed tissue macrophages, as well as few specialized endothelial cells present in the bone marrow, spleen, and lymph node, is called the reticular endothelial system. The reticular endothelial system is essentially the system of macrophages that guard various important organs and nodes present in our body. These include tissue macrophages in the skin and subcutaneous tissues, those are known as histiocytes. So essentially, we know that the skin is impregnable to invading pathogens and bacteria. But when the skin is ruptured due to a cut, there are tissue macrophages present just underneath the skin and the subcutaneous tissue, which are ready for any bacteria that comes to invade through the cut itself. Next, there are macrophages that are present in lymph nodes. So the lymph nodes and the lymphatic system is a system via which bacteria and foreign particles can travel. In case of an infection that is occurring in our tissues, the bacteria in most cases cannot travel through the bloodstream itself. It has to travel through the lymphatic system, and upon traveling via the lymphatic system, it encounters the lymph nodes, where there is a large amount of macrophages present which phagocytose these bacteria and eliminate them. Next, there are macrophages called Kupffer cells, which are present in the liver sinusoids, as we can see right here. Now, we can see right here. The Kupffer cells are specialized macrophages which are the resident population of the liver itself. And as we know, the bacteria have a favorite route of entry via our stomach and the digestive tract. Liver is all the blood and the nutrients that come from the stomach first must pass through the liver, where there is a huge amount of macrophages which are present and are very efficient at killing any foreign particles and pathogens. These macrophages are the Kupffer cells, and they engulf and phagocytize any bacteria that comes from via the digestive tract and tries to enter the blood. Next, there are alveolar macrophages in the lungs. So our lungs and the alveoli are also exposed to the external environment, and foreign organisms can also enter our body through our airway. These alveoli are in turn guarded by macrophages as well, which can then destroy any invading foreign organisms that come via the air path through our lungs into our alveoli. And finally, there are macrophages present in the spleen and bone marrow. The spleen is the lymphoid organ, and bone marrow is as the spleen. A large reticular has a large reticular network that, which means there is a network of tissues present inside both the spleen and the bone marrow. And a variety of macrophages are stuck inside the spleen and bone marrow, and wait for various bacteria invading pathogens in order to phagocytose them. This is all that we had on the topic of white blood cells or leukocytes. We have covered the leukocytes themselves as well as the monocyte macrophage or the reticular endothelial system. Next, we move on to immunity. Immunity can be divided into two parts. One is known as one kind of immunity is rather directed towards the general processes, which stops every kind of bacteria and pathogens that try to enter our body. These processes are directed in a general manner than towards a specific organisms, and this is known as innate immunity. Phagocytosis of bacteria and white blood cells. Phagocytosis of bacteria and white blood cells and everything before that we have already discussed fall into this category. Destruction of swallowed organisms by the acid secretions in the stomach and digestive enzymes also fall into this category. Resistance of the skin to invasions by organisms also stops any kind of foreign organisms pass through, and is also a general mechanism of preventing bacteria or other foreign organism to pass into our blood, is also falls under the innate immunity category. And finally, presence in the blood of certain chemicals and compounds that attach to foreign organisms or toxins and destroy them without looking at specialized organisms, but generally destroying all of them present, is also part of the innate immunity. But there is a second type of immunity that we will discuss today, which is the acquired immunity. In addition to its generalized innate immunity, the human body has the ability to develop extremely powerful specific immunity against individual invading agents such as lethal bacteria, virus, toxins, and even foreign tissues from other animals. This is called acquired or adaptive immunity. Acquired immunity is caused by special immune system that forms antibodies or activated lymphocytes that attack and destroy specific invading organism or toxins. What we mean by specific invading organisms and toxins is, say, you have a SARS-CoV-2 infection of a particular strain. Our body will prepare its acquired immune system to be primed towards that strain of SARS-CoV-2, and next time you have an infection, our body will generate various cells such as the T lymphocytes and also antibodies, which fight specifically against the SARS-CoV-2 virus. Those complement of cells and antibodies cannot target any other bacteria or organisms and are specific towards that strain of SARS-CoV-2. This is what we mean by acquired immunity. So, what are the basics of acquired immunity? Acquired immunity are divided into two types: humoral or antibody-based or B-cell immunity, and cell-mediated or T-cell immunity. In case of B-cell immunity, the body develops circulating antibodies, which are globulin molecules in the blood plasma that are capable of attacking and destroying invading agents. Antibodies are also generated by the B lymphocytes. Antibodies are generated by the B lymphocytes. Next, we have cell-mediated or T-cell immunity. The second type of acquired immunity is achieved through formations of large numbers of activated lymphocytes that are specifically crafted in the lymph nodes to destroy the foreign agents. These activated lymphocytes are the T lymphocytes, and thus this type of immunity is known as T-cell immunity. Both types of immunity are initiated by antigens. So what exactly are antigens? Each toxin or each type of organisms almost always contains one or more specific chemical compounds in its makeup that are different from all other compounds. So essentially, every organism or toxin has a fingerprint which can be detected by our immune system. In general, these are proteins or large polysaccharides, and it is they that they initiate the acquired immunity. These substances are known as the antigens. So we look a bit more closely into the origin of T lymphocytes and B lymphocytes, as well as the type of immunity they provide. So both the T lymphocytes and B lymphocytes are also made from the hematopoietic stem cells, but when they are released into the blood, the immature lymphocytes first home into either the thymus or the bone marrow, in which they mature. Depending if they travel to the thymus or the bone marrow, they are named the T cells or the B cells respectively. After maturing in the thymus and the bone marrow, these immature T and B lymphocytes reach a particular lymph node. These mature T and B lymphocytes reach a particular lymph node and reside there and wait for till they encounter a particular antigen. When T lymphocytes encounter antigen, they become activated T lymphocytes and start the cell-mediated immunity pathway. But in case of B lymphocytes, when they encounter antigen, they differentiate to form plasma cells, which generate antibodies at a huge rate and are capable of, and these antibodies are capable of directly or in activating the humoral immunity pathway. But a question comes that how these T lymphocytes and B lymphocytes are able to detect a particular antigen. What happens is that millions of different types of preformed B lymphocytes and T lymphocytes that are capable of forming highly specific antibodies or T cell receptors against a particular specialized foreign organism have been stored in the lymph tissue. Each of these preformed lymphocytes is capable of forming only one type of antibody or one type of T cell receptor with a single type of specificity. And only the specific type of antigen with which it can react can activate it. Once activated, these active lymphocytes reproduce at a wild, tremendous rate and create a number of clones or daughter lymphocytes. Each clone is again responsible to only a single type of antigen, and therefore when an appropriate antigen comes along, it immediately attaches to the antibody or the T cell receptor present in the cell membrane, and this leads to activation of that particular path of immunity, be it cell-mediated immunity or humoral immunity. So next we will look into a bit more detail about the type of immunity provided by both the T lymphocytes and the B lymphocytes, and we'll start by antibody formation by B lymphocytes. Before exposure to a specific antigen, the clones of B lymphocytes remain dormant in the lymphoid tissue. On being presented an antigen by the macrophages, the B lymphocyte specific for the antigen immediately enlarge and take the appearance of lymphoblasts. So the daughter lymphoblasts further differentiate to form plasmablasts followed by plasma cells. These mature plasma cells produce gamma globulin antibodies at an extremely rapid rate, about 2000 molecules per second. As said, the antibodies are essentially gamma globulins called immunoglobulins. All immunoglobulins are composed of combinations of a light chain, as shown right here, light chain, and a heavy chain. And there are two of each heavy chains and light chains. Further, the antibody whole structure is divided into a variable portion and a constant portion. The variable portion of the light and the heavy chain together form the antigen binding pocket, which binds to the particular foreign antigen. So we look into what the mechanism of action of antibodies really is. Antibodies act mainly in two ways to protect the body against invading organisms, which is by direct attack on the invader or by activation of the complement system. So we will learn about the complementation system just after this. But first, let us start at the mechanism of direct action of antibodies. So when antibodies try to attack the foreign object directly, these mechanisms include first agglutinization, in which multiple large particles with antigens on their surface, such as bacteria or red blood cells, are bound together into a clump, such as we can see right here. So big molecule, big particle, which has a number of antigens, has multiple antibodies bound to it, and antibodies cause this whole large amount of foreign particles to agglutinize and form clumps, and thus inactivating them. Next, next mechanism is precipitation, in which the molecular complex of the soluble antigens such as tetanus toxin and antibody becomes so large that it is rendered insoluble and precipitates out of the blood. And as it precipitates out of the blood, it is rendered inactive and cannot react or produce some deleterious effect on the organism. Next is neutralization, in which antibodies cover toxic sites in the antigenic agent. So if an antigenic agent, such as say a protein, has an active site via which it acts, the antibodies can be specific to the active site itself. And when the antibody binds to the active site, the foreign particle cannot bind to its normal target and produce any ill effects, and thus the foreign particle is neutralized. Finally, lysis. In some potent antibodies, they are capable of directly attacking the membranes of the cellular agents and cause rupture of the particular foreign cell, destroying it. This was the mechanism of direct action of antibodies. Next, we move on to the complement system. So what is a complement system? Complement is a connective term that describes a system of about 20 proteins, many of which are enzyme precursors. Many of which are enzyme precursors. The enzyme precursors are normally inactive, but they can be activated mainly by the so-called classic pathway. So these enzyme precursors are normally cannot be activated, but after action of other enzymes can act and produce a cascade of events, which then produces the action of the complement system. The classic pathway is initiated by the antigen-antibody reaction. That is, when an antibody binds to an antigen, a specific reactive site on the constant portion of the antibody becomes uncovered or activated, and this in turn binds directly to the C1 molecule, that is the starting molecule of the classic pathway of the complement system, and setting into motion a cascade of sequential reactions that end up creating various consequences, which we'll just look at. But first, we'll look into how exactly the complement system is activated. The antigen antibody complex acts on the first complement C1, activating it. C1 catalyzes the reaction between C4 and C2, forming the complex C42 and releasing another particle, which is a C4A. C42 then cleaves C3 to form the active C3b, which in turn cleaves C5, which forms the active C5b, which in turn catalyzes the reaction between C6 and C7 to form C5B67, which finally catalyzes the reaction between C8 and C9, forming the C5B6789, which is the lysis complex of the complement system that is finally formed. Various products in separate stages of the complement system also have separate functions, such as opsonization of bacteria, activation of mast cells and basophils, chemostaxis of white blood cells, and finally, this final lysis complex can also lyse foreign particles. So let us look into the role of the complement system. The major first role of the complement system is opsonization and phagocytosis. So what is opsonization? Opsonization is the process by which a particular object is made more attractive to phagocytes such as neutrophil and macrophages by the complement system. So the more this particular phenomenon is also shown by antibodies, which when bind to a particular foreign object can opsonize it or make it more attractive to the macrophages and neutrophils to eat it. So essentially making it more tasty or making it look most tasty to the to the particular phagocytes. So activation of phagocytes, which engulf bacteria to which the antigen antibody complex is attached, is one of the roles of the complement system. The second role is lysis. The lytic complex, which is a combination of multiple complement factors designated C5b6789, has the direct effect of rupturing cell membranes of bacteria or other invading organisms. Next, agglutination. The complement products also change the surface of the invading organism, causing them to adhere to each other and promoting agglutination. They also take part in neutralization of viruses. The complement system and other complement products can act on structures of some viruses and render them non-virulent. Chemotaxis. The complement system can cause large numbers of phagocytes to migrate into the tissue area adjacent to the antigenic agent and thus act as a chemotactic source. Thus, they call the various phagocytes to their location and have them engulf the foreign particles. Next, they also take part in activation of mast cells and basophils. The complement system causes the release of histamine, heparin, and other substances which increase the local blood flow, increase the leakage of fluid and plasma proteins into the tissue, and other local tissue reactions that help inactivate and immobilize the antigenic agents. And finally, the complement system can have inflammatory effects on the local tissue. That is, the increased blood flow, increased capillary leakage of proteins, and prevent movement of invading organisms to other tissues. This is how the complement system acts in order to fight against invading organisms, and this was the indirect pathway via which antibody and antigen antibody reaction can finally act against the invading organisms. Next, we move on to T lymphocytes and cell-mediated immunity. On exposure to proper antigens as presented by the adjacent macrophages, the lymphocytes of a particular specific lymphocyte clone proliferate and release large number of activated, specifically reactive T cells. So these are the activated T cells that we are talking about, that then start proliferating at a huge rate and directly act on the particular foreign invading cells. Although B lymphocytes recognize intact antigens, T lymphocytes respond to antigens only when they are bound to specific molecules called MHC proteins or major histopathological or major histopathic complex proteins on the surface of antigen presenting cells. Sorry, the T lymphocytes bind to antigens only when they are bound to specific molecules called major histocompatibility complex proteins on the surface of the antigen presenting cells. The three major types of antigen presenting cells are macrophages, B lymphocytes, and dendritic cells. Interaction of cell adhesion proteins is critical in permitting the T cells to bind to the antigen presenting cells long enough to become activated. So what happens is, as an antigen presenting cell has a MHC protein on its surface. The MHC protein binds to the foreign protein or the foreign antigen after processing it. The processed antigen is then recognized by the T cell receptor, and upon binding to this MHC and foreign protein complex, the T cell receptor allows the T cell to become activated. The cell adhesion proteins, which make sure that the antigen presenting cell and the T cell are bound together while this activation process is going on, favors the activation of T cells. So, there is a quick part of difference between B lymphocytes and T lymphocytes. B lymphocytes can detect directly the various antigens that are floating around, but T cells need to have been presented the particular antigen by an antigen presenting cell. So we look into the types and functions of various T cells. The first type of T cell is the helper T cell, which helps the immune system, as the name suggests, by releasing lymphokines. So what are lymphokines? Lymphokines stimulate the growth and proliferation of various cytotoxic T cells. They also stimulate the B cell growth and differentiation, activate the macrophage system, and provide positive feedback to helper T cells to produce more helper T cells. So essentially, it is a positive regulator of the whole immune system and helps the whole system geared towards fighting an infection. Next, we have the cytotoxic T cells, which are the T cells that directly attack foreign cells and kill them. They secrete perforins, which are cell membrane digesting enzymes, and other digestive and cytotoxic enzymes towards a particular foreign organism in order to destroy it. And finally, we have the suppressor T cells, which provide a negative feedback to the immune system. So the suppressor cells prevent excessive immune reaction. They produce a negative feedback to the immune system and prevent the immune system of the host to attack its own cells and promotes immune tolerance. These are the types and functions of the various T cells that are present in our body. So this is a diagrammatic representation of how the helper T cells and the cytotoxic T cells act. So first we look into the helper T cells. The first thing that happens is that there is a preprocessor unit, which can be macrophages or dendritic cells, which actually bind to the antigen and process it. And the process antigen is presented to the helper T cells with the help of MHC proteins. The helper T cells, which is specific to the particular antigen, binds to the processed antigen and produces lymphokines. And these lymphokines are the major path via which the helper T cells help modulate the immune system. The first thing they do is they help the proliferation and activation of cytotoxic T cells and suppressor T cells. Next, they allow the proliferation and differentiation of B cells so that they produce plasma cells, which in turn produce antibodies. Finally, they also act directly on the preprocessor areas such as the macrophage system and activate them and allow a large number of macrophages to be activated to attack the infectious agents. And finally, the lymphokines also act directly on the helper T cells themselves to promote their proliferation and growth and allow more helper T cells to be activated to release infections. This was how lymphokines work. Next, we have the cytotoxic T cells. The cytotoxic T cells, as you can see right here, can attach to the antigens present on the foreign cell And upon binding to the foreign cell, it releases various cytotoxic and after a while allow and after a while these digestive enzymes finally destroy the particular infectious cell and thus eliminating the infection. This is how the T cells and the helper T cells and the cytotoxic T cell act. Finally, we look into immune tolerance and autoimmune diseases. The immune mechanism of our body normally recognizes a person's own tissues as being distinctive from bacteria and viruses. And the person's immune system forms few antibodies or activated T cells but against its own antigens. Experiments have shown that specific immature lymphocytes in the thymus, when exposed to a strong antigen, become lymphoblastic, proliferate considerably, and then combine with the stimulating antigen. This in turn causes the cell to destroy by the thymic epithelial cells before they can migrate to and colonize the total body lymphoid tissue. Thus, in case while the particular immature lymphocyte was maturing, it encounters a self antigen, it will start proliferating and produces antibodies and activated T cells against that self antigen. But while it is still present inside the thymus or the bone marrow, the thymic epithelial cells start destroying these self-activating T cells, and thus prevent these T cells to get into our bloodstream and attack our own antigens, and thus they play a role in immune tolerance. All or most of these clones of the lymphocytes that are specific to damaged body's own tissues are self-destroyed because of their continual exposure to body antigens. As the thymic or the thymus is directly connected to the blood flow, most of the self-antigens flow right through the thymus and the developing T cells. So if a particular T cell clone has the ability to bind to a self-antigen, it starts proliferating, is identified, and then destroyed by the thymus. Thus, the final complement of T cells that is released in the blood are ordinarily such that they cannot act against any self-antigen. So we move on to autoimmune diseases, or the result of the fact that our body's immune system does indeed recognize self-antigens and attack. Sometimes people lose their immune tolerance of their own tissues. It occurs after destruction of body's own tissues and release of considerable amount of self-antigens, presumably which then causes the T cells or antibodies to be activated against that self-antigen. Several specific diseases can trigger autoimmunity. So these autoimmune diseases occur when our immune system catches hold of self-antigens and start building antibodies and immune response against it. One of the examples can include rheumatoid fever, in which body becomes immunized against tissues in joint. So what happens is that our knee joint and other joint are encapsulated, isolated systems. So the maturing T cells have never encountered antigens present inside the knee joint itself. But in case there is an injury and some antigens do get released from the knee joint because of the injury, the T cells will now recognize these antigens as foreign particles, as it has not seen them ever before. And the immune system starts getting geared towards acting against this seemingly foreign particle. And hence, we have the disease known as rheumatoid fever, in which the immune system starts acting towards our joint tissues. The other type includes glomerulonephritis, in which the immune system attacks the basement membrane of the glomeruli. Glomeruli are tissues present in the kidney themselves, and these are attacked during glomerulonephritis. And finally, we have myasthenia gravis, in which immunity develops against acetylcholine receptor proteins in the neuromuscular junction. So we have already seen the acetylcholine receptors present in neuromuscular junction, which receive the acetylcholine signal from the presynaptic neuron, which commands the muscle to contract via delivering the action potential. But we have also seen that if the acetylcholine based channels are inactivated somehow, the muscle is unable to contract. This happens during myasthenia gravis, in which our immune system gains starts attacking acetylcholine channels present in our neuromuscular junctions. Once these acetylcholine channels are inactivated, even though there is a signal from the presynaptic neuron, the muscle cannot respond to the acetylcholine signal and thus cannot contract, and hence myasthenia gravis causes paralysis. So this was all I had today for you. Thus we have covered in this particular chapter various blood cell types such as RBCs and WBCs, their structure and function, and also looked at acquired and innate immunity. I thank you for listening to this video.